All right. Hi, everyone. Our group is doing a presentation on proteins. So in a protein, there are multiple levels of hierarchy, and Arthur and I are going to be demonstrating what happens at the most basic level, or the monomers of proteins.
Okay, so first we're going to show off a dehydration reaction where water leaves the amino acids. For example, I have serine and threonine here. They're both polar molecules. So you would take off the OH of the carboxyl group and take off the hydrogen. That hydrogen, I knew that. The hydrogen off of the amino group, put those together and make water. And then you combine the carbon to the nitrogen to make a polypeptide bond, and that's during a dehydration reaction.
All right, and I'm going to be demonstrating a dehydration reaction between alanine and glycine. Basically, these two amino acids, they are nonpolar because, as you can see, the R group of this amino acid is just hydrogen, which is nonpolar, and equally shares electrons with everything else. Alanine has a methyl group, which is CH3 at the bottom. And as you all know, methyl is unreactive, and it only serves as like a structural purposes. And so basically what happens is you take the hydroxide off of the carboxyl group, and you take one hydrogen off of the amino group. This creates a water molecule, and this bonds into a polypeptide, a peptide bond. And now, in order to make the next higher level of
polypeptide bond,
there's going to be a polypeptide bond.
So you would just
bring this over here. And so basically what happens, again, you take the hydroxide off, and then you
create water, and then you would connect once again the carbon and the nitrogen to make a polypeptide bond. And so this is, in a sense, the primary structure of a protein, except the primary structure, Eddie will get into it later, is much longer. This is just more specific. And it just shows you how they bond between, and there would be water surrounding it.
So for our primary structure, we chose the protein collagen, collagen 1. And this is a structural protein inside your body, and over 90% of collagen is actually collagen 1. So the primary structure of a protein is just the amino acids linked together. There isn't actually any structure or any shape to it. It's just like a list of amino acids. So over here, a few things to note with collagen 1's primary structure is you can see there's a yellow gumdrop every 3 gumdrops. So basically, yellow represents glycine. And the amino acid structure of collagen 1 is very unique in that it has a glycine XY structure, so it has a glycine every 3 amino acids. So basically, these are all linked with dehydration reactions, and by dehydrating all the amino acids, we combine them into a long chain of amino acids. And this is just a small portion of the full amino acid, full polypeptide chain.
So as Eddie mentioned, this is only a portion of the amino acid chain, and I'm going to be representing each of the 3 amino acids chains in collagen with these 3 colored Twizzlers. And then I'm going to be talking about the secondary structure. The secondary structure of a protein has 2 parts. It has the triple helix and the beta-plated sheet. This is a triple helix. It has the 3 alpha chains, and it is connected through hydrogen bondings. And in a protein, they twist around like this. They form like this. And the next structure, as you can see, is the beta-plated sheet. The beta-plated sheet is an undulating lateral hydrogen bonding between the 3 chains. It has a specific structure, which we will mention later. And then these 2 structures form the tertiary structure, because when they combine, they form the tertiary structure. And as you can see, I have 2 of them, because when 2 of the tertiary structures combine, you form a quaternary structure, which is the final form of a protein, which Eddie will get into.
Okay. And the quaternary structure is actually, for collagen, it's a fibrous protein. So it's not really, so this is a fibrous protein, and it's kind of long, and it's very long. And basically, collagen bonds together to form fibrils and fibers. And as you can see here, these are each of the colors here represent one polypeptide chain. So since there's a triple helix, these 3 is one. 3 of these colors is one triple helix. And basically, each polypeptide chain is bonded together using, they're covalently cross-linked. So basically, they're bonded together with covalent bonds. And as you can see, these starbursts in between there are representing the covalent bonds between each of the chains. And the way this is bonded is there's another protein called lysyl oxidase that basically it modifies the amino groups of lysine to get them to get hydroxyl lysine and lysine to covalently bond together. And it oxidizes the amino group actually, and it allows it to covalently bond. But on the other hand here, this is not actually collagen, but for demonstration purposes, we have a globular polypeptide here. And as you can see, there are four different polypeptide chains that are exactly the same, at least as similar as I can make them. And in the middle, there's a green starburst that basically shows the interactions that they have between each other. And one of the other important things that proteins can do is denature. For example, when the temperature is very high, they will lose their shape. So, for example, with this globular quaternary structure, if the temperature gets too high, then the proteins might, then each of the polypeptide chains might come off, and then each of these would unravel. And since structure determines function, this doesn't have the same structure as before, so it wouldn't be able to function the same way, even though the primary structure is still exactly the same.